Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. My name is Ayunda. You can call me Sonia. My ID number is 20031103. I'm from class Biology Education of the Biology major State University of Padang. And now I will presentation about cell division. That is mitosis. Mitosis. Before you talk about mitosis, you must know about meaning of mitosis. And mitosis and mitosis is the process of cell division in which the chromosomes in the nucleus split into two identical sets of chromosomes. In mitosis, the two daughter cells have the same number of chromosomes as the parent cells. Okay, next mitosis stage. In mitosis stage, it start with prophase, and then next metaphase, next anaphase, and the last telophase. And in the telophase, there are cytokinase and synthase and coenzymes. Okay, number one is prophase. Mitosis has begun when the unwatched chromosomes began to coalesce. During the first stage of mitosis, called prophase, the small cylindrical bodies become very important when present, and you can see that in figure 1.1. They help organize much of what is about to happen. The cat just outside the nucleus, an animal says, though not present in most higher plants, the centrioles began to move to opposite ends of the cell. As this happens, the DNA, which is the primary constituent of the chromosomes, recoils. During this process, the chromosomes become more distant, while the nuclei become less distinct. This is what called a prophase. It is in the nuclei that ribosome production occurs. During mitosis, when the chromosomes condense, ribosome production ceases. When unraveled, the chromosomes interact with their surrounding medium. However, for cell division to occur, the already replicated chromosomes must be pulled apart. Yet this can't happen until the chromosomes have condensed, making it possible for cell division to proceed. And once recoiled, the chromosomes become XFs. The XR comes of two identical chromatids, each held to the other by a single chromosome centromere. You can see at figure 1.2, one of these chromatids was copied from the other during interphase. When replication occurred, while the chromosomes are equal, the nuclear and nuclear envelope are disappearing. And at the same time, a series of microtubules are forming the spindle. The spindle is the characteristic grouping of microtubules that occurs during nuclear division. It helps to alike and move the chromosomes. During the early stage of spindle composition, microtubules radiate around each centriole, creating formations collectively known as the esters. Although most higher plants do not have centrioles, they still develop spindle fibers at prophase, but esters do not form. The spindle fibers are the microtubules that together constitute the mesocytic apparatus called the spindle. During prophase, the chromosomes move toward the middle or the equator of the cell. By the end of prophase, the nuclear membrane is no longer visible and has broken down. This is figure 1.2. Next, number 2, metaphase. Metaphase lasts only as long as all the chromosomes remain lined up along the equator. The centromeres have divided in two, each is attached to one of the two corresponding chromosomes from the pair. The individual chromosomes are called homologs, and together both chromosomes in each pair are called homologous chromosomes. Next, 3. Anaphase. Anaphase begins when the two complete sets of chromosomes start moving toward opposite ends of the spindle. Each chromosome appears to be dragged along by its centromere, which is attached to spindle fiber. The division of cytoplasms, or cytokinesis, begins at the end of anaphase. Next, the last, telophase. Telophase is the last stage of mitosis. This is when cytoplasm separates in two parts of the cell, while the cell's plasma membrane pitches in from both sides, creating two distinct cells. While this is occurring, each of chromosomes reaches its respective pole, where the nuclear membrane forms, enclosing the chromosomes. The chromosomes then uncoil, while the nucleus repairs. Upon completion of cytokinesis, a new centrole is made. And in telophase, there are number 1, cytokinesis. Cytokinesis, the division of the cytoplasms, usually begins in late anaphase and is complete in telophase. In animal cells, cytokinesis begins with the formation of an incision, or cleavage furrow, which form all the way around the equatorial region, becoming deeper and deeper until it cuts right through, leaving two distinct daughter cells. Plant cells also have a plasma membrane that divides in the same manner. In addition, plant cells have a rigid cell wall that can form a cleavage furrow. Instead, a cell plate forms at the equatorial region, and rather than moving from the outside in, the cell plate begins forming inside and grows toward the periphery. And the last, syncytia and choanocytes. As already discussed, the nuclear membrane forms during telophase and cytokinesis occurs. However, this does not happen in all types of cells. Some lesions have cells that undergo mitosis divisions. They are not followed by cytokinesis, so the cell ends up containing many nuclei. When nuclear division is not followed by cytokinesis, the result in animals is called a syncytium, and in plants it is a choanocytes. It's enough for me. Thank you for attention. Assalamualaikum warahmatullahi wabarakatuh.